Ioanna and Vaz joining us from Birmingham. Ioanna works in chemical engineering and Vaz in neurology. Vaz is actually at the University of Nottingham. So the two of them are going to fill this next 10-minute slot with a bit of food science and a bit of neurological science, I believe.
I'm a postdoctoral researcher at School of Chemical Engineering at the University of Birmingham. And one of the things I'm focusing on in my research is the sort of structures that are involved in foods. But before we go further, I just wanted to clarify a bit what goes into a standard emulsion. So some of you may have heard the word emulsion. I don't know if today you mentioned at all the word emulsion, but especially if you're a fan of cooking shows, or you may have also seen it on a fancy menu. But if you haven't heard of this word before, please don't worry. It only takes the opening of your fridge or your freezer to probably find in there many foods that are considered emulsions. For example, the butter, the margarine, milk, mayonnaise, the ice cream, some sauces, salad dressings, and so on. So an emulsion, if we're actually talking about liquids, is essentially the result of combining two liquids, for example, oil and water, that normally do not mix in a way that makes them stay mixed. So today I'm going to take the example of salad dressing, which has oil and vinegar as two of its main ingredients. And I will show you how to prepare a successful homemade salad dressing and one that doesn't look that great. So I have here, I hope you can see it in this small pot, some sunflower oil, and I'm going to be adding to this. I'm going to be adding some vinegar. So yeah, as we expected, we see right away that these two ingredients do not mix and they form these two separate layers with the vinegar at the bottom and the oil floating on the top. So one way to get these to mix together, if your intention is to make a salad dressing, is to shake them together. And this will break down one of the ingredients into tiny droplets. So cap goes back on. And we shake it briefly. So there you go. We have made an emulsion. But I promise you, this will not stay like this forever. It will stay like this only for a short period of time. And eventually, because I don't really want to bore you with this, it will take about 10-15 minutes. They will start separating out and they will look like this. But that's not what we want really in our salad dressing, right? We want one that's all mixed together. So a properly made vinaigrette has a thicker consistency than either the vinegar or the oil. So the trick is to add to this a third ingredient, which is an emulsifier, we call it. Chris spoke briefly about this. It will keep the two liquids together almost permanently. And some common emulsifiers you can find in your kitchen are mayonnaise and also honey, which is what I will be using today. So what I will do is that I will add these emulsifiers into the mix here and in another one I made here to test which one is the best in the sense that it holds the two liquids together best. So while I'm actually adding my emulsifiers, if you want to make a prediction, which one you think will work better among these two, please feel free. So let me try. I'm adding first a scoop from the mayonnaise here. Okay, that should be enough. Right, and I will add also a scoop of the honey, which is like a teaspoon. It's a bit trickier. Okay, so I will actually pass them on now to Vas for some agitation, which is a fancy word for shaking, really.
Like
so,
that, yeah?
Yeah, so he will shake them for us. So yeah, mayonnaise is a common emulsifier. Why? Because it has eggs in it, and the egg yolk contains a functional lipid which is called lecithin. And again, if you want to go more technical, the emulsifier consists of molecules of a water-loving nature and a fat-loving nature. And in this way, the emulsifier acts as a binding agent between the fat and water, in that the molecule would bind these two together. So this is what comes out of the shaking. We made some emulsions, and we're gonna let them settle for a bit. And these are some emulsions that I prepared like 10-15 minutes ago. This is the one with honey, and this is the one with mayo. And as you can see, for the mayo, it looks fully mixed. It has changed also a bit, I don't know if that's clear, but it has changed a bit of color. So mayonnaise has mixed very well, and there's no oil thin layer at the top.
Yeah.
On the other hand, that's what happened with honey. So it did not work well at all. So oil is at the top, and the vinegar has all set to the bottom. So it did not prove to be a very good emulsifier. An alternative would be to try adding as an emulsifier the mustard. So I've made an emulsion before. Again, I've let it to set for about 10-15 minutes. It did not work perfectly. It's a pretty good emulsifier, but as you can see, after 10-15 minutes, there's like a thin oil layer, a tiny bit of separation at the top. So that will be my next suggestion if you want to try something. So I'm taking now my most successful preparation, which is the one with the mayo, because the last thing would be to make sure it has a nice taste. So I'm gonna add a bit of that. If you want to add some flavorings or spices, that's fine. And I'm gonna leave it to Vassilis to try it and to start his own session. Thank you very much indeed, and I hope you enjoyed today. Thank you.
Thank you very much. That was fantastic. Vas, if we can interrupt your feeding, you can tell us a little bit about some of the work that you do. Thank you so much.
Yeah, sure. Thank you, Adrian. Yeah, now I feel stronger after tasting this nice vinegar. Hi again, everybody. My name is Vassilis. I'm a neuroscientist. And as part of my research, I'm interested in understanding how our brain works, and in particular, how we see and hear stimuli, how we see and hear the world around us. So to crack on, I would like you to have a look on the following classic visual illusions. In this one, I would like you to think a little bit or talk out loud, or even write here on the chat, which of the two orange circles you think is bigger. I imagine that most of you might think, well, the right one is bigger than the left one, right? However, I'm afraid that they have the exact same size. I know this is not intuitive at all, but the reason they look the way they do is because the left one is surrounded by bigger circles compared to the right one, and as a result, it looks smaller by comparison. Then in the next one here, I would like you to think a little bit which of the two central rectangulars is darker. And again, I would imagine that you might think that the bottom one is darker, right? However, I have to say they have the exact same shade of gray. And again, the reason they appear like that is because they are surrounded by different colors, right? By a darker gray surrounds the top one, and the lighter gray surrounds the bottom one. Finally, here, as you may have figured out already, these two lines have the exact same length. Although I'm sure initially you might think, you may thought that the bottom one is longer, right? The reason they appear like that, although they have the exact same length, is because of the particular arrangement in their arrowheads at the end, at the edge of the lines. So all these illusions, actually, what they demonstrate is that our perception, our brain, our perceptual system, our visual system, is a bit, is a bit plastic. Is a bit, it gets influenced by what a visual stimulus is surrounded by, and it has some sort of relativity. It's a constructive process, right? It's not like the camera at the bottom of phones. So in my work, in order to find out how the brain works while we are seeing things like that, we are using a complicated machine called an MRI scanner. I'm sure most of you may be familiar with an MRI machine. It's a very popular tool in medicine. All big hospitals have at least one MRI scanner nowadays. So in my work, I'm using this to collect data by running experiments, then more experiments and more experiments, and then I go to my office and I analyze those data, and after more and more analysis and complicated analysis, then brain appears, as I promised. This is a tool that helps us see what's going on inside our heads. And finally, I would like you to have a look on some real data, some data that I have collected from people to get an impression, get an estimation of what's going on in the brain when we see faces, and how face perception might be different from place perception when we see places in the brain. So here it is how it looks. So at the moment you are seeing me talking, for example, it's very likely that something like that is happening in your brain. These sort of areas are activated now that you see a particular face. And then in my final slide, I would like to have a look on how we hear things. Then now you are hearing me talking, for example, various sounds, are getting out of my mouth, low frequency sounds, high frequency sounds. And by using again the same technique in the lab, we can visualize, we can have an estimation on how the low frequency sounds and the high frequency sounds are represented on the surface of our brain. And by doing something like that, we can compare how this looks in normal hearing to how it changes in situations like hearing loss, for example, and help patients by designing the right treatment for them. So that's it for me. I hope you found that interesting. Thanks very much.
Thank you very much, Vas. Thank you very much, Ioanna. That was really good. It's great that you could join us. And thank you for taking time out on your Saturday to speak with us live today. And
Thanks very much.
Catch up with you soon.
Thank you, Adrian.
Thank you.
Bye.
Bye.
Bye.